Good morning everyone and welcome to my YouTube channel. So here today we are going to be looking at the parts of a prokaryote. For example, when we are looking at the bacteria. So what are some of the parts that make up the bacteria? So you may look at the diagram. We have the capsule. We have the capsule. So we have the cytoplasm here, which is the liquid part. We have the plasma membrane, which is more like our cell membrane if we look at the animal cell and the plant cell. We also have the cell which is made up of peptidoglycan or murein. We also have the ribosomes. We also have the plasmids. We also have the pili. We have the bacterial flagella. Not forgetting our nucleoid, which is our circular DNA, and the plasmid. So now let's dive deeper into the details of what the functions of these are. So let's look at them. So what we have here is a simplified diagram to help us understand the parts and the structures. So firstly, we are going to talk about the flagella. The flagella is more like the tail of the bacteria or the prokaryotes. So this one is used for movement. So as it produces the movement of the tail, it brings about the movement of the entire organism through certain rotations. We are going to look at the proton motive force in another video. So now, the flagellum's function is to help the bacteria or the prokaryote in locomotion, which is just movement from one point to another. So now we are going to neglect the vacuole, which is not predominantly present in the bacteria. So now we have the plasma membrane. As we know, the plasma membrane, it controls what leaves and enters the cell. Then when we look at the ring DNA molecule, this one, the ring DNA molecule, which we were calling nucleoid, this has got the function of storing genetic information for the formation and the passing down of genetic information from one organism to the other. If you see that another bacteria is formed and it has got the same genetic information, this is because of this DNA. It is more like kept preserved to form another bacteria after asexual reproduction. The other thing that we have is the cytoplasm, which is the site of chemical reactions. The other thing is the mesosomes. The mesosomes functions is to act as a site of respiration. This is where the enzymes that aid in respiration are mostly found. Then when we look at the plasmids has got the function of more like creating antibacterial resistance. So the bacteria's resistance is mostly created by the plasmid. So by definition, the plasmid is a circular DNA that is not part of the main central DNA. So as you can see, this is the main central DNA of the bacteria, which is just suspended in the cytoplasm. Then the plasmid is not part of the main circular DNA, but has got the function of creating antibacterial resistance. So the ability of a bacteria to resist antibodies comes from the plasmids. Then we also have the capsule. So the capsule is more like responsible for creating slime. So the slime that the capsule creates helps it not to be engulfed by antibodies. When we say engulfed, we simply mean to say to be swallowed. So due to the slime that it produces, it's more like it's slippery for the bacteria, for the antibodies to swallow it. So this slime helps it being prevented from being engulfed. Then the other thing is that when we look at the pili, which are these structures, the pili have got the function of helping the bacteria to be able to attach to host cells. So the bacteria most of the times will be required to attach to some host cell. So this is helped by the pili. So these help the bacteria attach to other organisms, or rather the hosts. So not forgetting our cell wall, which is made of peptidoglycan or murein. So this has got the function of protecting the bacteria and also giving the shape of the bacteria and maintaining the cells more like the cell from undergoing lysis, meaning to say the cell of the bacteria from breaking down upon being exposed to a lot of water. So bacteria sometimes may more like gain a lot of water. So due to this, the cell wall prevents the bacteria from bursting when it gains a lot of water. So these are the functions. Let's just write a summary. So on our summary, we have the flagellum, which are long tail-like structures that help in the movement of the prokaryotes through its rotations. Now we are going to dive deeper into the flagellum later in the next video when we explain how the flagellum brings about movements through the proton motive force. The other thing is the ring DNA molecule, which we will be calling the nucleoid. So this is the main circular DNA that carries essential genes that are important in the control of growth, metabolism, and reproduction. So now this ensures genetic continuity. What we simply mean is that if you have one bacteria, it will be able to split to form another bacteria having the same DNA. So this is extremely important as it enables genetic continuity, meaning to say there is the same DNA that the mother had as well as the daughter cells. The other thing is that the cell wall, which we talked about, being made up of peptidoglycan, which is also known as murein, it helps in the giving of the prokaryotes its shape. Now, not only that, it also prevents what we call lysis, which is the breaking down whenever the bacteria gains a lot of water, which is one of the functions of the cell wall, among others. The other parts that we have talked about, it is the capsule, which produces slime. Now slime is important to make the bacteria to be slippery, thereby preventing antibodies from engulfing them. When we say engulfing, it's more like they are being swallowed. So this slime prevents that thing from happening. We also looked at plasmids, which are the circular DNA, which is not part of the main circular DNA. So we have the main circular DNA, which we were calling nucleoid, but the plasmid, it helps in the creation of antibody resistance. So I said in this video, the plasmids help by creating antibody resistors. So this helps the bacteria to survive. In other words, we can say that it helps the bacteria to be able to adapt against any hostile environment that may be hostile to it. We also talked about the pili, which helps in the attachment of the bacteria to its host. So the bacteria needs to attach to its host for survival and continuity. We also talked about the mesosomes. The mesosomes are the site of respiratory enzymes, which is also responsible in the formation of ATP. ATP is the energy that can be used in various processes such as locomotion, or rather movement from one point to another. We also talked about the cytoplasm, which is the site of reaction. So these were the parts. We have the cytoplasm, which is the liquid part. We also have the nucleoid, which is the main circular DNA. We also have now the plasmids, which are not part of the circular DNA but also carry genetic information, such as the one that is responsible for antibacterial resistance, antibody resistance. We also have the pili, which are responsible for attachment. We also have the bacterial flagellum, which helps in movement. We have the capsule, which helps in the creation of a slime. We also have the cell wall, which gives the bacteria its shape, and also prevents lysis. We also have the plasma membrane, which controls what enters and leaves the cell. We also have the mesosome, which is the infolding of the cell membrane. When we say infolding, it means to say, as you can see, this is just the cell membrane which has folded inside to enter more like the cytoplasm. So it is an infolding of this. It is the infolding of the plasma membrane, which has got the function of the site of the site of enzyme action and also ATP formation. We also have one important thing that we forgot, the ribosomes, which are helpful in the creation of proteins. So the ribosomes found in prokaryotes are the 70S ribosomes, which you must know. So the 70S helps in the creation of proteins. So that is very, very important. So the ribosomes found in the prokaryotes are called the 70S. So that's that for this video. Thank you very much for watching. Hope you would like, share and subscribe for more. So kindly like so that my video may be recommended to a broader audience.